Okay, today we're going to do the second part of cell division. What I want to go over today is an actual diagram of the cell cycle, okay? So all of you have a handout that I distributed for writing on. This is the cell cycle. If you are watching this at home, I will draw it out for you. And we're going to fill it in so we know what each portion of the cell cycle is representing, okay? So the first thing is you just need a big old circle. If you're at your desk, you already have this. I'm just drawing it out. Okay. All right, so far so good. All right, there it is, the cell cycle. So let's get to labeling it. Fletcher, what is the majority of the cell cycle? What phase? Definitely, especially in us mammals, it can be up to 90% of your entire cell cycle. Now, a moment ago, I mentioned the Tampa Bay Lightning. I'm sure you guys have been to a game or two. In a hockey game, you have these things called intermission. In football, it's kind of like halftime, but there's two intermissions. Intermission is a break. You generally do two things during your break. You recover from the previous period and you get ready for the next period. Well, intermission, interphase, same idea. The cell just went through a division, so it is recovering from that division. And it is also getting ready for the next division. So if you go to a Lightning game this year, when you see the players go in the locker room, they're catching their breath, probably get some water, and they're getting ready for the next period, okay? So it's a little bit of both. So we're going to highlight this entire boundary between A, B, and C all the way around. All of that is interphase. So I'm going to really spell it out here. I, N, T, E, R, P, H, A, S, E. Interphase. All of that. Now, interphase has three main parts. You have part A, B, and C. These have names. Part A is known as G1. In your notes from yesterday, you have what G1 is, and I'll reiterate it now. G1 is normal behavior. The cell is doing its thing, but it's also getting ready. And the first thing it's going to do is it is going to replicate all of its organelles. You need to double it. If you had 5 mitochondria, well, now you have 10. If you had 12 Golgi apparatus, now you have 24. You're doubling everything because you were going to make 2 cells. Every cell needs a full copy. The next stage, part B, is known as the S phase or S stage. This stage is where the DNA replicates. See if you all remember a number I said from yesterday. We are humans. We're Homo sapien. How many chromosomes does our species have? 46. If you've ever heard of the DNA heredity thing you can mail in called 23andMe, I mean, what's 46 divided by 2? 23. You get 23 chromosomes from your mother. You get 23 chromosomes from your father. That's 46. So we have 23 pairs. If you had 23 pairs of shoes, you technically have 46 shoes. And then we had the G2. G2 is part C. This is where you make your final preparations. You're getting ready for the big act, division. So to recap, G1 is the cell has just gone through a division. So it's kind of like, in a manner of speaking, catching its breath. But it's also going to be performing normally, doing the things the cell does. And it's also going to be doubling the organelles. The next is S, as in synthesis. You're going to be making more DNA. In fact, you're going to double the amount of DNA. And then G2 is your final preparation. Okay? So if interphase is all of that, what is part D, the minority of the cell cycle, Will? That is called the M phase. Very good. M phase is going to be all of part D. Now, the majority of part D in the M phase is called mitosis. I know you've heard that one before. Mitosis is made of 4 main stages. What is the first stage of mitosis? Prophase. Next, metaphase. Next, anaphase. And finally, telophase. Good. Those are the 4 stages. And then mitosis is nuclear division. The nucleus is actually dividing into 2 nuclei. What is that process where the entire cell divides from 1 cell into 2 distinct cells? It's part of M phase after mitosis. Check your notes from yesterday. Cytokinesis. I don't know why it's glitching. Anyway, that's cytokinesis. And that's it, guys. That is the entire cell cycle. Now, this is on your mastering. As the guy that puts together your mastering and handpicks the questions that are on your mastering, this is on there. But there's actually one last portion I want to get to. It's something very important. They're called checkpoints. These checkpoints try to ensure that DNA is not damaged and that you are not passing on mutations to future cells. Unfortunately, or fortunately, depending on how you look at it, these checkpoints are not foolproof. Some mutations are very good. I have a mutation that runs in my family where I don't have wisdom teeth. Wisdom teeth is an extra set of teeth. It's like your 3rd molars that can sometimes crash into your other teeth, and they're called impacted, and you have to get them surgically removed. Anybody had their wisdom teeth out yet? Anybody know that's coming in their future? So I don't have to worry about that. My dad doesn't have wisdom teeth. His brother doesn't have wisdom teeth. Apparently, some of my dad's side of the family. I inherited that mutation. Another mutation I inherited that probably originated about 10,000 years ago or so, blue eyes. If you have blue eyes, you're likely from Northern Europe. Your ancestry is from that region. If you have green eyes, you're a mutant. If you're blue eyes, you're a mutant. If you don't have dark skin, you're a mutant. If you didn't know, all humans originated from Africa, darker skin. If you have lighter toned skin, you're a mutant. We are all mutants in our own way. When I look at Cooper's face, I just have like this facial recognition technology in my brain. We all have it where, oh, that's Cooper. And oh, that's Fletcher. I'm not going to say, hey, Cooper, hey, Fletcher. I know who's who. Well, the reason why he looks different than Fletcher and Fletcher looks different than Cooper is because he has mutations that no one else has. His nose is a different shape. His face is a different shape, different eyes, all of it. We are all mutants in our own way. Some mutations are just neutral. They don't give you a benefit, but they don't give you a handicap either. Some are really good. I'm happy I don't have to have my wisdom teeth ripped out of my mouth. And then some mutations are bad, like cancer. Cancer is a mutation. It's a pretty nasty mutation. And it does happen. Raise your hand if you know someone that's had cancer or know someone that has suffered from it. Majority of you. Almost all of us know someone that suffered from cancer. Hopefully you guys never will encounter it, but statistically, some of you in this classroom will get cancer. You have safeguards built into the cell cycle to try to stop it. But the fact that I have blue eyes or Cooper has light skin or someone might have blonde hair is proof that these checkpoints are not 100% foolproof. So the checkpoints are found in 3 locations. The first one is at the end of G1. This is checkpoint number 1. The whole purpose of these checkpoints is to make sure the DNA is not damaged. But these checkpoints are not 100% foolproof. Nothing in this world is. The second checkpoint comes at the end of G2. And the final checkpoint is at the end of metaphase. We call this the M checkpoint. So the whole point of these checkpoints is it's like a proofreader. If you've ever had to write a book report or you've had to write an essay, you can go to spell checker on a Word application. Or what I used to do is I'd ask, my dad used to be a teacher, so I said, Dad, can you read this? I think I'm clear of typos. In my English class, we would get a point off for every typo. So we had to make sure no typos, no grammatical errors. So I said, Dad, can you read this? I checked it. I'm pretty sure I've got all the typos. Can you check? And he hands it back to me and I missed like 5 things. I'm like, wait a minute, I proofread this myself. I know I have it all right. He found 5 things. So even though we sometimes think that we proofread it and we got all the mistakes, we tend to miss one. It's nature. You're human. Oops. Blue eyes. Oops. That's a mutation. Wasn't meant to happen. Blonde hair. Oops. That's a mutation. Wasn't meant to happen. No wisdom teeth. Oops. Anybody else have a cool mutation or an odd mutation in this room you don't know about, or maybe you do? What do you have, Natalia? What? I think I've heard of that. Interesting. Yeah.
I have 3 extra bones in both of my feet.
That's a mutation. Absolutely. Most people do. Most people have deviated septums. If you kind of get out of track here. So if you're listening on YouTube, try this now. You want to know if you have a deviated septum. A septum is something that divides 2 halves. Like this wall is a septum. So here's how you check. Close one nostril, blow out the other. And do the opposite. Did one nostril blow out better than the other? You have a deviated septum. I do. I broke my nose 3 times. I definitely have a deviated septum. If you blew out one nostril better than the other, you have a It's not straight. Most people have a deviated septum, so it's not unnormal. So long as you can breathe, you're good. All right, guys. Well, anyway, while I'm just talking about this whole thing with cancer, let me tell you really quickly what cancer is, okay? Because we hear about it all the time. Next month is like Breast Cancer Awareness Month. You're going to see football players and hockey players and baseball players wearing pink. You know, what's up with that? So you don't have to write any of this down. I really want you to just pay attention. So let's say here's a stomach where we put all of our food. And let's imagine that there's a little spot right there of abnormal cells. These cells are abnormal because they don't know when to stop growing. Every one of your cells should have a gene that tells them, hit the brakes, stop growing. These cells don't have that. It's like a car without brakes. So over time, this area of infected, mutated cells gets bigger and it spreads. And before long, the stomach cannot work properly. Do we need our stomachs to survive? Yes, it's a vital organ. So if you develop cancer on a vital organ, here's what basically happens. You have a huge mass of cells that are just weird, not right cells. They inhibit your organs from working properly, and your organs begin to shut down. Here's an analogy I like to teach. If you've had me before, you've heard this one. Your parents gift you a car for high school. They say, "Here, Merry Christmas, here's a car." The car has rust on the engine, but the engine runs. It runs fine, but it has rust on the engine. So you don't pay the rust any mind. As time goes on, that rust starts to spread across the engine. It starts to get into vital components of the engine. The engine still works, maybe not as great, but it still works. And so over time, the rust continues to spread over the engine. And then one day you go to turn on your car won't start. You pop the hood, the engine is covered in rust. Nothing works anymore. The pistons won't pump, the spark plugs won't fire, transmission shot, nothing works. The rust is cancer. The engine is a vital organ. If the vital organ can't work, it shuts down, which means your life could shut down. Now, cancer also spreads. My aunt died of breast cancer. Now, breasts are not a vital organ. You can live without them. But it spreads. It's called metastasis. When I taught EP Bio, Morgan, you probably remember this. You had to properly remember this word and pronounce it. Some people just said, call it meta cancer. Metastasis means that it spreads. And in my aunt, her breast cancer metastasized to her liver, her brain, and her spinal cord. Do we need those to live? Yes. And that's what she died of. She by the end of her life, she didn't know who she was or where she was or who we were, because it got into her brain. And it really, really messed her up. It just it was like rust, just covering the brain, preventing it from working properly. That's what cancer does. It spreads and it just shuts down the organs. It keeps them from working. So when you hear cancer, that's really what it does. These checkpoints try to stop it, but these checkpoints are not foolproof. Okay, so there is your diagram. Now what I want you to do is I want you to get out your packets and get out some new notes, because you will probably be able to work on your packets while you're doing these notes. We're going to go over the stages of mitosis: prophase, metaphase, anaphase, telophase, and then cytokinesis, and that'll be it for today. Chemo tries to poison the cells so that they die. But you're also poisoning your body. So that's why people get really sick. Chemo is just a bunch of chemicals. That's where the chemo means. And then the other one is radiation, where they try to just burn it away, like nuke it, microwave it. Okay. Here we go, guys. You may want to have your packets out and also your notes out. The first one is going to be interphase. Yes or no? Interphase is part of mitosis. No, it is not. Interphase is not part of mitosis. During interphase, the cell is working fine. It's behaving normally. It's recovering from the previous division. The nucleus is intact. The DNA is decompressed or decondensed, I mean. And I'll give you guys a few labels here. This is the nucleus. What's inside your nucleus? DNA. And this whole cell or this whole structure is the cell. Okay, so the cell is just chilling. It's doing its thing. It is, however, recovering from the previous division and preparing for the next division. We're going to call this the parent cell. When I say parent, I don't mean your mother or your father. I mean the original cell, the cell that's going to be copied. And yes, it is raining outside if you have sea lunch or sixth lunch, whatever they call it. It's going to rain now. I don't want it to rain during the game. Yes, Natalia. Say what? Mitosis. Ready? Yeah. You guys are going to draw a lot of circles here today and a lot of structures inside. Okay, zooming out. After interphase, what's the first phase of mitosis? Prophase. Now, as you guys saw from your notes yesterday, prophase has the most going on of any of the phases of mitosis. So what a lot of people do is they break up prophase into 3 parts. And that's what we're going to do. So the first part of prophase, we're going to call early prophase. In early prophase, the DNA has now condensed into chromosomes. The centrioles move to opposite ends of the cell. These little guys right here are centrioles. You have 2 pairs now. And those little stick figure looking structures inside the cell, those are your chromosomes. How many chromosomes does this cell that I drew have? How many do you see? Okay, Cooper says 6. You're right and wrong. Kinda. Once you guys are writing, I'm going to tell you what the real number is and why. It's very important that you hear that, but I know you're drawing right now, so I'll wait a second. Okay, Cooper, you're a male, right? Do you know what your sex chromosomes are? XY, very good. So I want you all to stop writing for a moment. I want you just to watch this. It's going to be really important. Cooper said that there are 6 chromosomes within that cell, within that nucleus. He's right and he's wrong. Here's what I mean. So let's say here's Cooper's X chromosome, and here's his Y chromosome. He's a male, X and Y. Every single one of his cells from head to toe has X and Y in it, as well as the other 44. All 46 chromosomes are in there. When Cooper's cell is getting ready to divide, each new cell is going to need a copy of that DNA. So here's what happens. The DNA doubles. So now he has 2 X's. Oops. And he has 2 Y's. And then during division, he's going to get an X and Y over here and an X and Y over here. so that each new cell gets a full copy of the DNA. So when I say he's right and he's wrong, here's what I mean. There are 6 chromosomes, but there's 2 copies of 6 chromosomes. You don't need to write this down, but you got 1, 1, 2, 2, 3, 3, 4, 4, 5, 5, 6, and 6. You have 2 copies of each chromosome. This person would, or this organism would have 6 chromosomes. And now they just have 2 copies of them. Okay. Let's move on to the next one. Prophase. During prophase, nucleus is going away. It's eroding like a sandcastle on the beach during high tide. It's going away. And the spindle fibers, which are kind of like little ropes, are starting to be assembled by the centrioles. The centrioles are organelles that help the cell divide. Those lines that are emitting from the centrioles are called spindle fibers. They're made of the same stuff as the cytoskeleton. You can see the little red fragments I have. That's what's left of the nucleus. It's going away. Yes. I left out a chromosome, didn't I? Oops. There we go. Anyone in here have 6th lunch? Obviously, outdoor eating is closed. You can go in the auditorium or the cafeteria. Can't be sitting in the rain. Okay, we're about done with prophase. Let's move on to the final step of prophase. There's 2 names that would be acceptable: late prophase or pro-metaphase. Notice that the nucleus is history. It's gone. The chromosomes are now free. But they're being captured by the spindle fibers, and they're going to start to be pushed towards the cell's midline, or we call it the cell equator. But when you think of the Earth's equator, you go from left to right. The cell equator is going to be from top to bottom, but it really doesn't matter. Just along the midline. Yes? I can't hear you, Natalia. You speak very softly. I had no idea what time picture retakes are. I have no clue. I thought it was during your English period. Is it? There you go. Can you get that? Natalia, you're signing out. This will be on YouTube, okay? Okay, we done with pro-metaphase, late prophase, same thing? Couple more seconds. Ready? All right, let's go to metaphase. Meta means middle. Look where all the chromosomes lined up. Right down the middle. Here's a question that I ask every year, and the answer is very obvious. Sometimes here's my hypothesis is that kids think that the answer is so obvious I must be messing with them, like it's a trick question. It's not a trick question. The answer is as obvious as you may think. If I want to split the cell in half, of all places, why do the chromosomes line up along the middle? To divide evenly. That's it. E word. Even, equal, to divide evenly. That equator is called the metaphase plate. It's just the midline down the cell where all the chromosomes line up. So as Aiden said, and several others of you said, you can divide it evenly. Okay. Next is anaphase. Ana in Latin means away. Look at how the chromosomes are being pulled away from each other. So 6 are going left and 6 are going right. This is going to be towards the new cell. The spindle fibers are going to pull the chromosomes apart to opposite poles. A pole means an end, one end or the other end. And you can also see the cell is starting to stretch. Remember all this, Zoe? Is everybody done with metaphase? Okay, after anaphase is telophase. 2 new nuclei begin to reform. The chromosomes are going to decondense back into just general DNA called chromatin. And there's something called a cleavage furrow, which is going to start to split the cells in half. But look very closely. Look at telophase. Are the cells actually physically split yet? Not yet. They're getting there, but they're not separated yet. This is called a cleavage furrow, where they're kind of pinched across to be separated. I'm a little fast, guys, because I'm only allowed 40 minutes on Zoom, or I won't save the recording. We're almost done. Is everybody done with anaphase? Okay, after telophase, we have cytokinesis, and that is the end. We now have 2 separate daughter cells. These daughter cells are identical to each other, and they are identical to the original parent cell. There have now been 2 cells successfully formed from one parent original cell. Cytokinesis is not part of mitosis, but it is part of M phase. M phase is mitosis and cytokinesis. The cells are now physically separated. And that's all. Well, I'll put that back up.